Hello, hello. Good evening, everyone, of computational biology for your final year projects. Computational biology to be an interactive session, which I plan that it is going to be for 3 days. However, it might not actually go to that length, but I want you to just pay attention so that as we go on, you'll be able to learn one or two things. Don't forget, I don't want to take your time because time is fast spent. And what we actually want to cover is going to be um, a bit, is going to be a bit more of our project. So don't forget that the use of bioinformatics is also called computational biology because we are going to make use of computer for your project. That's why they call it computational biology. So we are going to see some of these as we go on. These three cohort group, the first group is a vaccine group, which we are very conversant with. Vaccine group, the use of in silico or the use of computational technique for vaccine design. That is group one under this arrangement. Then the second group, which is going to be antimicrobial resistance group, which we fondly call AMR group. This is also the use of computational biology for predicting antimicrobial resistance. We can see that antimicrobial resistance in microbiology has been a very serious public health problem because most of the drugs that or antibiotics that they use in controlling them, they are no longer effective, and that's the need for us to be able to use or make use of a computer in order to predict, in order to estimate, in order to be able to uh, detect antimicrobial resistance gene. So it is going to help us to be able to do that to the genetic level. Then the last one is going to be um, drug design, and that's the reason why we actually need to be very conversant with what we are going to need. Now, just like I said, um, this lecture is going to provide a comprehensive details for us for the purpose of using in silico vaccine design, antimicrobial design, which I call AMR, and the drug design using computational methods. So, and the purpose of this is to be able to use the modern day technique, and that is the use of a computer, that we can make use of it as a complementary to our traditional method, traditional laboratory method, or we can use computational biology throughout for the design. So we need to know that. Now, before we'll be able to go on, in these three groups, I want to introduce us to some of the definitions of key terms, most of the terminologies that you will come across them. So I want to briefly start with mentioning them. The first one is in silico. In silico, we are very used to the word in silico. This refers to processes or experiments that is conducted on computer using computer simulation or uh, computational method as a kind of a complementary method to our traditional laboratory method. Don't forget, we also have another group, which is the laboratory group. So the laboratory group is the one we are used with. It is called the traditional method of project execution or carrying out projects. So computational method is the use of a computer, that is in silico. Then you also come across the word antigen. The word antigen. Antigen are foreign substances, foreign compounds, or foreign elements. It can be in form of micromolecules that will elicit immune response. So what we are saying in essence is that antigen, they are foreign. Apart from being foreign, they must also have the ability to trigger an immune response. If they cannot trigger an immune response, that means they are not antigen. They cannot be used to design what? To design vaccine. And I also want you to know, I told us the other day we met, I told us that vaccine is just substances or molecules that will prepare our immune system for the danger ahead. So that is a vaccine. So when we are now making use of computers, if you are making use of computer in order to be able to design vaccine, that is called in silico. So then also, what do we mean by AMR, that is antimicrobial resistance? This is the ability of microorganism. It could be bacteria, it could be um, fungi, it could be viruses, it could be other pathogens. It could be other pathogens that resist, that word, please underline that word, that resist or prevent uh, or render useless the antimicrobial agents. For example, if the antimicrobial agent has been a kind of chemicals, so that has been used to to control them in the past, but now they are no longer effective. They are called, that is a process that is called, or a phenomenon that is called antimicrobial resistance. Then some of us in drug design, and vaccine, you will also come across what is called molecular docking. What do we mean by molecular docking? Docking, docking, molecular docking. So that is um, the use of is a computational technique. It is a computational technique that is used to predict the correct orientation. So what do we mean by orientation? Let us say, let us assume, okay, like padlock, key and padlock. Now, if the key is not in the right orientation, or if the key is not in the right orientation, that means it might not be able to open the door. The same thing with docking. So when we are talking of docking, that is a computational technique that they use in predicting the preferred orientation for a molecule. So we will come across that. Then we also come across in drugs, you also come across what is known as pharmacokinetics. Pharmacokinetics. That is the study of absorption, distribution, metabolism, and excretion within the body. So it is like you want to study, you want to know the journey of a drug or certain drugs from the entry, that is the entry point, to the exit point. That is what is called pharmacokinetics. And if we are talking about pharmacodynamics, so pharmacodynamics, this is the study of biochemical and physiological effects of drug on the body. So we will also come across that. Now, let us move straight to the first group, which is In Silico Vaccine Design group. So what do we uh, understand by that, and what are the prospects and potential of in silico drug development? That is, if we make use of this technique, if we make use of this method, so what are going to be our potential or the prospects in that? Number one, there's going to be accelerated development. Accelerated development. COVID-19 has been a very good example for us. COVID-19 came in 2019, and because of the use of a computational technique or computational tools, we were able to get vaccine to control COVID-19 as far back as 2020. That is less than 1 year after. So, if we make the prospects of in silico design of vaccine is that it is going to lead to accelerated development. Then number two, it is going to be cost effective. It's going to save a lot of costs. In fact, the group of the laboratory group, I can tell you authoritatively that what we have spent on getting consumables so far, if we are spending that on computational technique, it is not going to be as much as that, so to say. So the use of computational technique is more cost effective. Then apart from that, it will also remove hazard completely. It will remove hazard, the danger of mishandling live viruses, or let me say live microorganisms, the danger of inhalation, the danger of getting yourself infected because you are working with live biological agents. So what we are saying in essence is just that it is more cost effective. Then apart from that, it is precise. It is more precise. It is towards the goal. You move from the your destination. You will see, before we go on, we must have designed an algorithm that we are going to follow from day one down to the last day, and it is going to be precise. You know where you are going. You just go there straight. You don't need to try to run anything like trial. You want to practice this, you want to practice that, you want to do. No, it is more precise. Then it is also more personal. It is personal, and that's why they call it you and your personal computer, you and your laptop. You interact more with your laptop. You can be alone. You might not necessarily work in group. That means it is personal. So another prospect or another advantage of in silico vaccine design is that it's more precise and it is also personal. Then the last one, which I want you to know, is that pandemic preparedness. It will make you to be able to prepare. Once you are being able to design your vaccine very well using your computational technique, it is already preparing you for what in case there is any pandemic. I'm very, very sure, had it been there was the usage of computational technique before COVID-19. In fact, before COVID-19 will come, people will have designed what? They will have designed vaccine for that. So that is that. Now, what are the application of this technique? Application of in silico vaccine design group. What are the application? Number one, it will help us in epitope prediction. I mentioned something that we call epitope. What do we mean by epitope? Epitopes are uh, they are antigenic region. They are antigenic region that is capable or that are capable of eliciting an immune response. So I've just told you the other day that if you have lengths of protein like 20, like 200 amino acids, when you look at the amino acid or when you look at the whole protein, it is not the whole protein that is going to be used for vaccine design. There are certain segments which are more antigenic than the other, and that is what we call um, epitope. Epitope. Then I also told you. Now let us move to the algorithm. What do we mean by algorithm? Algorithm that you are going to make use of if you are working in vaccine design group. Algorithm is like a recipe. If you want to prepare any delicious food, you will need some recipe, you will need some ingredients, you will need that you first of this is the first one you are going to add. Before you will add this, it is going to be sequential. So recipe is, you can use recipe to explain what algorithm are. So what are those algorithm? That is, where are you going to start? The first basic algorithm that you are going to need, the first one is data collection and pre-processing. Data collection and pre-processing, that is number one procedure. The data collection and pre-processing. What do we mean by data processing? The first one is that you collect the genomic sequences of pathogens. All these pathogens you collect, you are going to collect their sequences, the sequences of their protein. Uh, I told you that it's not all proteins that they use for virulence. It is not all protein. So you are going to make use of, you are going to retrieve, so you can call it retriever of pathogen sequences, retriever. So that's the first one, and you will now pre-process it. Part of the processing you are going to use include, you are going to determine whether it can be used as vaccine. You will at that point you will test for localization. Where are they located? Are they located externally? Are they located internally? Where are they located? You are going to test that. Then apart from that, you will run blast, you will run similarity checks. Is part of the first process you must be familiar with. You will check the similarity, what you are going to blast those sequences together. Why do we need to blast? You will blast in order to know whether there is what you call homology. That is similarity between, there must not be any similarity between the vaccine substance that you want to use and that of human. So what we are saying is so that there won't be any cross reactivity. There won't be any cross reactivity. So you must make sure that you blast, you check for homology, you check whether they are subcellular localized, that is, are they localized in the cytoplasmic matrix, or they are then apart from that, immediately after that, you will check all those. We are going to give you some of the databases that you are going to use in order to be able to check, to pre-process after retrieval. After you must have retrieved all those sequences, you are going to check for similarity. That is similarity check. you check, you are going to blast them. Apart from that, you will clean that data. That's the first thing you need to do. You need to clean that data. When you clean the data, then you can proceed to the any other one. Okay, without wasting your time, let me just run through the algorithm, the list of algorithm we are going to use for vaccine design. Uh, Vaccine design. Okay, let's go there. Okay, okay. The first one, which I call the data collection and pre-processing, is what I have told you. The next one is the one that is going to help you, epitope prediction. We are going to predict all those amino acids on the sequences that you have. We are going to check whether the combination of that amino acid can be used as epitope. I've explained what epitopes are for you. Okay, so that's going to be the next one after that. And you are going to make use of some software like IEDB, that is Immune Epitope Database. IEDB, Immune Epitope Database. Or we can make use of NetMHC to predict potential epitopes. Potential epitopes. That is, we want to predict the antigenic epitope, antigenic prediction. So we are going to make use of the next thing is molecular docking. I've mentioned molecular docking for you. Molecular docking, this one is going to allow you to get the correct orientation, the correct binding angle. Is going to be so when after you must have gotten your protein, um structure of your protein, you will start to know the inclination, the angle at which it is going to be well fitted. So that is number 3. Okay. Then number 4, that is vaccine candidate selection. So that is where the point at which you are going to design your vaccine. That is the level. It's on level 4 that you are going to now construct your vaccine. So your vaccine, you must have constructed it. Now, under this molecular docking, okay, under the epitope prediction, there are uh, so many software that we are going to make use of. So there are so many. This one can take you like 1 month, 4 weeks, 5 weeks, doing that continuously. So epitope prediction. We'll talk of interferon, the role of interferon, the role of interleukin, and so on and so forth. So this epitope prediction is a very long and very broad process, as well as a molecular docking. We are going to make use of that. We have different websites or database that we can make use of in that. For example, we'll make use of Autodoc Final. Then apart from that, there are other like Galaxy Refine. We are going to refine it under Galaxy, and so on and so forth like that. So you are going to see all this. So and we now move to vaccine candidate selection. Vaccine candidate selection, that is the level you are going to construct. You must have gotten your vaccine candidates at that point in time. You must have gotten the compound. So by the time you have gotten that compound, you now do the the secondary structure construction, the use of PyMol, the use of others like others. There are many of them that we are going to make use of under the vaccine candidate selection. You know all this because this not the first time the first time in bioinformatics. Then the last one is going to be simulation and optimization. Simulation and optimization. There are also different software that you can use for this. For example, we can make use of a GROMAX to optimize the vaccine design. We make use of we are going to simulate the immune system of the host, so that it's going to predict, it's going to act. We want to see how the vaccine is going to react in the immune system of the host. So this level is the level you are going to make use of GROMAS and some others. Like there are different types of this. You are going to make use of that. Okay. Now, summarily, I've told you that we are going to start with you start. Once you have made up your mind, you start. The next one is that you collect data, and that is the pathogen genome. You must have the access to the whole genome of that pathogen. So you collect the data from there. So after that, you are going to identify conserved regions. Identify conserved regions. I told you, identification of conserved regions will definitely tell you that you can proceed, you can make use of it as to design vaccine candidates. Okay, so conserve region. After that, you will now go to predict epitope prediction. Epitope prediction. So after epitope prediction, the next one is which I have highlighted for you here. So, just like I said, I don't want to unnecessarily bother you for too many things. So, I want to really believe that one way or the other, you have learned one or two things about this. And don't forget that this is going to form the basis for all of you that are going to work on vaccine design group. We are going to follow this method, this procedure, in order for you to be able to do it. So once again, I really want to thank you. I have very few students that are online, very few of them online. So I want to really believe that for some of you that did not join now, I'm very sure that you'll be able to join me and do the assignment I've given you in the community, WhatsApp community group. I want you to drop it there so that we'll be able to check your assessment. So I want you to summarize what we have done in today's lecture and just put the summary in the comment section. So once again, I want to see you tomorrow morning, the same time, and then the same YouTube channel. Thank you, and God bless you.